I'm excited to be giving you this presentation. I'll just correct one thing from the intro. Before, my former chairs of those departments claim I'm stealing their titles. I held the chair, but I wasn't the chairman. So, anyway, it's nice to be here and to tell you a little bit about the work we've done that will connect back with some of the immune talks. I'm sure it connects up to mitochondrial function at some level. Maybe that's for the future. Let me share my screen. And there we go. Right, so I'll come to the immune system from a slightly different direction in this talk. And I'm a molecular pharmacologist, neurobiologist, and I focus on synaptic signaling and neural pathways underlying behavior. And in trying to identify some of the neural substrates for some of the behaviors that are affected in autism, we came upon a connection to the immune system that surprised me and that I think might be relevant for future pharmacotherapy. So let me tell you about it now. So many years ago now, my laboratory cloned the target of Prozac. This is the serotonin transporter, presynaptic protein that clears serotonin at synapses. And Prozac essentially blocks the activity of that protein and keeps serotonin levels high in the extracellular space. Now, given the role that this protein plays in antidepressant pharmacology and also in various aspects of serotonin signaling related to mood and mood disorders, that's kind of where I thought my career was going to head. And then one day, one of my postdoctoral fellows who was doing a developmental study came in with an image that was really striking and caused me to rethink that direction. And that was this. This is on the left, a cross-section through a mid-gestation mouse embryo. And you can see the eye, you can see the developing brain in this area. And what you see in these white dots is a reflection of gene expression. Each one of those dots is essentially a cell. This is gene expression for the serotonin transporter, or SERT, in this developing embryo. And this showed us that the serotonin transporter was being expressed far earlier than we had thought. We thought about this as a protein that works in the mature nervous system, clearing serotonin at essentially adult serotonin synapses. But here we were seeing it expressed very early in development, and this has been replicated now in many species, including primates. When we saw that, I began to think about developmental onset disorders that might have a serotonin link. And we've known for over 50 years that there are changes that occur in individuals with autism in terms of levels of serotonin in their blood. And I knew this. I had been trained a bit in psychopharmacology, and there are several other biomarkers related to serotonin that had been picked up in brain imaging studies. But on the right here, you see a distribution from a study. This has been replicated many times, where on the left, you see a normal range of serotonin levels in subjects, and this is in whole blood. And on the right, you see there's a subpopulation with a bump, indicating that they have greater than 2 standard deviations from the mean of high levels of serotonin in their blood. And this is this marker, while it still remains somewhat mysterious as to what it does in the periphery, what it's telling us in the periphery, we knew by this, we know now that most of the genes that control the production, the transport, like SERT of serotonin, or the serotonin receptors, are the same in the periphery as they are in the brain. So the idea being that if we could understand this biomarker a bit from a molecular level, perhaps we could understand something about autism. So that caught my eye, and I did a few studies before needing to step back and think a little bit about where this serotonin in the blood is coming from. And we know that the serotonin is stored in platelets. That's where 99.9% of blood serotonin is. Anything that's not in a platelet gets degraded. But platelets don't make serotonin. They actually take it up via the same protein that I had cloned before, the SERT protein. And they get it from gut cells called enterochromaffin cells. These are endocrine cells that release serotonin into the bloodstream. And then platelets just basically sequester it and hold on to it and travel around in the blood. And that serotonin can be re-released, and it helps with blood clotting. Well, so that means that elevated serotonin levels in the blood either mean more platelets, more serotonin production by the gut, more SERT activity on the platelet to take it up, or more storage in granules inside cells. So the fact that there is a SERT component in this story got me very interested. And I began to work with a genetics group at Vanderbilt headed by Jim Sutcliffe. And Jim was doing a linkage study looking for DNA elements that were selectively transmitted in a multiplex family paradigm to the children who had autism, but not to unaffected siblings. And in that effort, a locus was defined that overlay the serotonin transporter gene exactly where we said it should be. Well, we went right after that gene and identified in multiple individuals in this study multiple changes in the gene that changed the protein sequence. And the frequency with which we detected these changes was way beyond what we could have expected by going out and just collecting 300 families and looking, because these are very rare mutations. Now, rare mutations show up in many, many studies, but they may not do anything. And this was probably the most striking finding out of that work, which is they all do the same thing. And here you see a bar graph showing us serotonin uptake activity, and the wild type, or the reference transporter sequence, transports at this level of serotonin in transfected cells. And you see that each one of these variants transports more serotonin per unit time per cell. So here's a case, and Richard was alluding to sort of elevated function showing up in a phenotype. Well, here's another one. These aren't mutations that kill this protein. These are mutations that actually make it work more, that increase the activity, either by making more protein hang around longer so that there's an abundance of it, maybe putting more on the cell surface, or making it hyperactive. So we did studies on this to look, and what we found were that these three actually increase levels of protein on the cell surface. And this one, which happens to be the most common, it's at about a 1%, 1 to 2% level of frequency in North American Caucasian populations. And this was because it was as frequent as that, and because the surface level is actually lower, meaning if you divide activity by that, you actually get the most highly activated of the variants. We went after that one to study it more intensively. Now, having an activated transporter protein connected with basic research we were doing in the lab to try to understand how a neuron turns on and turns off its transporters. It can just sit it there all the time and take up serotonin as it comes in, but that's not how it works. These proteins shuttle in and out of the cell surface on demand, and when they get to the cell surface, they need to be activated. So we identified a two-step process that uses two enzymes, protein kinase G and P38 MAP kinase. PKG shoves these proteins into the surface on demand, and p38 MAP kinase activates them. Well, as I told you, this mutation leads to not elevated protein on the surface, maybe a little bit less, but this enzyme must be attacking this transporter much more actively and turning it on. At least that was the hypothesis. So we decided that one idea that made sense to us was that this protein was already in a state of activation and it couldn't be further activated. So we tested that by actually looking at the lymphoblasts from these subjects. Turns out that lymphocytes express small quantities of SERT protein, and you can measure the activity. And we got from our collection of individuals, these are subjects with autism, who we found some that had the wild type GLI amino acids that where the mutation was. Some had both alleles mutated and some had just one. And what you can see is that in the normal case, protein kinase G and p38 MAP kinase stimulate the activity of the transporter. But the ones that have 2 copies of this alanine mutation do not stimulate it whatsoever. Now I've normalized the starting point here in black without any drug added to stimulate those kinases to 100%. So you can see there's nothing further than 100%, but they start out active, higher activity to begin with. So this gave us the idea that this transporter is transporting so fast that it has no reserve and it should be sitting down lower. So normally it's hyperactive, and in the brain, that's not a good thing. It's going to be taking serotonin away from the synapse where it needs to act. So while they're hyperserotoninic in the blood, more serotonin in the blood, in the brain, this means reduced synaptic serotonin. And sometimes people get confused by that. Here's just an illustration. We've got SERT here, and with SERT activity going higher, we increase the amount of serotonin in the blood. But in here, in the synapse, where SERT is here to take up serotonin, when that activity goes up, this goes down, all right? So now the question is, how can we show that? That's just been in cells. So the way we decided to show that was to make a mouse and introduce that mutation. So we made the SERT ALA56 mouse. It's a knock-in. We just edited the genome, put that mutation in, and now we have a mouse that carries that variant. And we've studied males in our studies. We have some studies with females, but almost, all the data I'll show you is from males. And what we found after getting these animals and getting their blood, we had hyperserotonemia. This is the first time that biomarker had ever been reproduced in a genetic model with a gene change derived from studies in autism. So we were very, very excited. Does that really regulate SERT in the brain? Well, one way you can look at that is that you can put a little sensor in the brain that can measure the presence and decay, the disappearance of serotonin as it's being sucked away back into the neuron. And what you do with that sensor is insert it into the brain. It's like a hair, it's very thin. And next to it, you shoot a little serotonin. You see there's a pulse. That signal goes up. And then it's cleared, and you see in gray here, that's the wild type clearance. But over here in black, you see that the mutant animals are clearing serotonin much faster than the wild type are. So this meets the predictions that, in fact, in vivo, this mutation is causing too much serotonin clearance, and we suspected that would have behavioral consequences. Now, I don't have time to go through, happy to share the paper with anybody that writes me, but this has now been published, and what we found was that serotonin neurons have reduced functional capacity because of this mutation. There are changes in serotonin receptors, and the animals show problems with vocalization, social vocalizations. They show repetitive behavior. But I wanted to show you this behavior because it's a very robust one, and it was one that we were able to use to test for medication leads. So what this is called the tube test. It's sometimes used in studying dominance hierarchies, but really what it is an opportunity for two mice to meet in a tube they can't turn around in at the middle, and you score who backs out. Who doesn't want to stay there and to go nose to nose with this other mouse? And we score that as number of wins for the one that actually moves forward out of the tube. And what you can see is that the animals carry 2 copies of this alanine substitution. And I know that most of you are not molecular biologists by any means. And this is a very, very subtle change in the protein sequence. This is not something by looking at the DNA or looking at a protein sequence that any bioinformaticist would ever say is a damaging mutation, not at all. But look what it does. It causes these animals to withdraw from a social encounter. Now, I mentioned before that the change in this protein mimicked what happens when you activate P38 or PKG or P38 MAP kinase. So we're very focused on this P38 MAP kinase because the protein wasn't trafficking, it was activated. So we wanted to ask, is this transporter modulated by that kinase in the brain differently? And on this plot, what you can see is that this protein is carrying a tag that P38 MAP kinase adds to the transporter when it gets activated. So in other words, these transporters are what's called phosphorylated. And that means that they're in a state of constitutive activity mediated by this kinase. That's very important because now we have a drug target that's not the transporter. How would we want to reduce that activity? Well, we could give Prozac. We could give SERT antagonists like an SSRI. However, to now shut that down, we're going to go in and probably hit that protein and turn it off. That's not a good thing either. Why don't we turn down this enzyme and let the normal activity of the transporter be in place, but just not let it go sky high? Well, there weren't any inhibitors for the particular isoform of P38 MAP kinase that regulates SERT. There are 4 isoforms, and we needed a drug that could get in the brain, that was high affinity, and would block one of those 4 isoforms. And just as we had our mice and were ready to do these studies, a former colleague of mine at Vanderbilt, Marty Watterson, published work showing the development of a specific P38 alpha MAP kinase inhibitor. And it's called, the one I'll show you the work from is MW150. So this is a drug Marty is actually studying for its potential in treating neuroinflammatory disorders, including Alzheimer's disease and other neurodegenerative disorders. But we looked at it for a different reason. Could it help us with our animals? So we gave this drug once and we saw nothing. So of course he said, well, we just got to repeat dose this. So we gave it for a week and look at what happened. Whoops. I think I must have lost my screen. Okay, hang on just a second. Richard, are you still there? Unmute and tell me you can.
Yeah, I'm still here.
Okay, sorry about that. I have to find that talk again.
Yeah, no, just, are you able to find it?
Yeah, it'll be here in a sec. I'll get to it.
Okay, no problem. We'll give you a couple extra minutes.
Thank you. I don't know where it went. All right, it is. Let's back up. Share screen again. All right, so let's do the same thing. Okay. Got to open it directly here. Should be coming up. Okay, try one more time here. See if I can get through that slide. Okay. All right. So on the left, you see what I showed you before. In black is the normal clearance away of serotonin. And in red is what the SIRT-ALA56 mutant mouse does, clears it faster. You see on the right what's now happened after giving a once-a-day injection of this drug to these mice. The clearance now has dropped back down to be a normal rate. What about that social behavior? And we, in our paper, we use this drug to normalize a number of different things. I just wanted to show you this, because it's just particularly striking. So what you see now in the purple and the orange bars are what I showed you before, that when you have the ALA56 mouse up against a wild type mouse, it backs out. Well, after you give it MW150, there's no significant difference between that animal, that ALA56 animal, and the wild type mice. And these are adult mice. This is important. This says that whatever that mutation did, even though it's expressed very early in development, you must have to continue to have this significantly elevated clearance, or the system will reset. And I think that's very important, because we know we have a large unmet need of adult individuals on the spectrum. Now that's a drug administration. So all it does is it says that pathway is important, doesn't tell us where. So in order to show us that, in fact, that is working in a mechanism like I was describing to you, it was P38 acting in the serotonin neurons to regulate SIRT, we had to take a different approach. And that approach involved a genetic manipulation to knock out P38 alpha in serotonin neurons. We won't go through how we did that, but once we did it, you can see here as well that we have normalized back the social behavior deficit that we saw in the ALA56 mice, pointing to this pathway within serotonin neurons as a problem introduced by this mutation. Now, others have begun to see similar contributions that serotonin is making, particularly to social behavior. Here is a very nice paper from Jessica Walsh in Rob Malinka's lab at Stanford, using a different mouse model of autism. Here, the 16p12.1 locus duplication deletion syndrome. And in their case, they found deficits as well with serotonin signaling to a reward center called the nucleus accumbens. And if they just made these neurons fire more, get more serotonin out there, this overcame their social deficits. Sort of a complementary thing to what we had done. Well, I've talked about P38 MAP kinase, but I haven't mentioned that P38 MAP kinase is downstream of the receptor for major inflammatory cytokine IL1 beta. And we wondered in other studies that I don't have time to go through whether IL1 beta could activate SIRT by walking through this network. And the answer is yes. So IL1 beta will stimulate SIRT activity, and it requires activation of P38 MAP kinase, bringing us in contact with the inflammatory cytokine world. And today what we think is happening is that this mutation in the serotonin transporter is putting it in this activated state that we call Searchstar. And this is a state that the transporter normally gets into when the IL1 receptor activates P38 MAP kinase. The issue is that normally this receptor does this transiently, but the mutation puts it in that state all the time, and it's transporting at maximum velocity. Now, of course, if you have a lot of IL-1 beta around, you're also going to be pushing this transporter at a high rate. So you can get to the same endpoint by having a rare mutation here, or by having a chronic syndrome with IL-1 beta driving this transporter. Well, it's even more interesting than that. I have a colleague named Ning Kwan here at FAU, recruited here a couple of years ago. And what Ning is a specialist in is in studying the IL-1 receptor. And he made mice who have a tagged genetic locus that's fluorescent wherever that receptor is expressed. And what you can see here in red is that fluorescent signal for the IL-1 receptor. In green is a stain for serotonin. These are the serotonin neurons in the midbrain that I was showing you as in an embryo. This is an adult. What you can see is that there's quite a significant correspondence between the expression pattern of the IL-1 receptor and serotonin neurons, suggesting that in fact that these neurons use the IL-1 receptor all the time. They are there, they have this receptor on them and are paying attention to inflammatory states, or at least the presence of IL-1 beta. Now, these neurons we know innervate a few areas. The nucleus accumbens I mentioned before as an area that the Malinka lab had targeted to implicate serotonin signaling in social behavior. So we've been looking at this area, and what we have found is that when we elevate inflammatory cytokine IL-1 beta in the brain, we get an activation of neurons in this area. And if we take away the IL-1 receptor that's over here in these two bars, there is no stimulation through IL-1 beta that goes to the nucleus accumbens. So we think that we have now a simple logical circuit. We normally have serotonin neurons that are inhibiting a set of inhibitory neurons with their serotonin. When the serotonin transporter takes away that serotonin, these inhibitory neurons are not inhibited. And as a result, these neurons turn down this social engagement pathway mediated by glutamate secreting neurons in the nucleus accumbens. I think this is perhaps one of the labeled lines where immune signaling into the nervous system changes behavior. It hits serotonin neurons via the IL-1 beta signaling to an IL-1 receptor. That is going to shut down these neurons as well. The serotonin transporter here will keep the serotonin from stimulating these cells. These cells will now inhibit this social circuit, and we are going to get social withdrawal. I think this idea could be played out in some of the other areas that I'm looking at, but I wanted to point out that, again, this opens up the opportunity for pharmacotherapy. This transporter's regulatory kinase, not the transporter itself, using a drug like MW150, I think could be valuable as a therapy in autism. So with that, I'm going to stop, and just in orange are the fellows in the group and my collaborators that really did a lot of the work that I talked about, and happy to take questions. I hope there's time.
Yeah, I mean, we had a little glitch there and I went over a little bit. So yeah, there is, it looks like one question in the chat box, if you can look at that, or do you want me to read it?
Yeah, so it might be, let's see if I can, seeing the chat box is not.
Okay, I'll read it to you. It says,
yeah, go ahead.
Do you have any perspective on the likely impact of hypersert on other neurotransmitters? How do you test for SERT abnormalities if there's no SERT mutation on whole genome sequencing? In your view on ketamine as therapeutic? And finally, you may use your judgment in picking the questions.
Okay. All right. So first off, so SERT is selective for serotonin. So the impact on other neurotransmitters would be, from a mutation like this, would be indirect. Now, the serotonin and dopamine systems are in a very close collaborative circuit. For example, in nucleus accumbens, which is a reward center, and they modulate the normal appearance of rewarding stimuli and allow it to then move into the appropriate actions. So I think that there is a dopamine story buried in here as well. The other thing is that serotonin is a major modulator of GABA and glutamate neurons in the cortex. So it's very likely that the ultimate readout of serotonin's actions in the brain are through these cortical excitatory inhibitory networks that we hear a lot about in terms of their balance being disrupted in autism. How do you assay for it? Well, if you don't have one of these mutations, right, I would say, well, this is a story that's not just relevant for those people with those mutations, because they're not that common, okay? But I think that the biology tells us that this pathway is important in driving that particular behavior. And if that's a behavior that's altered in an individual, now we have another tool in our toolkit to see if we can bring something more appropriate back out. You can assay this in blood. You can blot the protein that's in platelets and assess the level of it. That's likely, in my mind, not to be as beneficial as knowing the activity of the protein. That has to be done in a different way by following the uptake of serotonin into the platelets, and that's done in a research laboratory. That's not something we do clinically.
Any in your view on ketamine at all?
There are a few studies suggesting that there is an intersection of ketamine on the serotonin system, but largely, the light right now is all focused around glutamate signaling pathway. So ketamine gets us into the depression side of the whole serotonin story. So I can't connect it in here for autism.
One of the things that's fascinating is the idea that the immune system modulates the social function like that, which evolutionarily, I guess, would seem to make sense, right? If you, at least to me, if you're a sick animal, let's say maybe because of some contagious illness, you'd want to go away from other people.
I think that's actually that's correct. Now, one of the other talks that we heard talked about in terms of I think it was Lisa Bollinger was talking about the normal actions of these immune molecules, and it's very likely that this is a signal. I mean, this protein is sitting there, this receptor for IL1 beta, probably doing stuff all the time. Maybe at lower levels of IL1 beta, it may have a developmental role. It may have a role in just normally modulating and fine tuning. Let's just go with social interactions. But then it can be corrupted by having a strong, constant inflammatory stimulus that really pushes the serotonin system over the edge, and you get this depletion phenomenon. So but I agree with you. I think that it does make ecological evolutionary sense. I think it's an ecologically responsible thing to be doing, just like you mentioned.